Thank you so much. Thank you so much for having me. Just to be clear, Build a Cell is not a competition for a life. We're not actually making artificial life, we're making fake life. There's a big difference that I'm about to talk about. Okay, so what do I want to talk about today is I realize this is not a very biological meeting. I deal with molecules. I'm actually a chemist by training, but I pretend to be a biologist now because that's where the money is these days. So we're making artificial chemical systems that resemble living cells. We're selling them as synthetic cells because that's what people want to see. But when you think about what I'm going to be showing, you really think about chemistry, because that's what we're doing. We're really putting together molecules in systems that have some, but not all properties of biological life. And that's kind of the goal. So just to give you a little bit of a background of what we're doing and why we're doing it. So this works. I swear this worked. Okay, so this works. Now can I switch my slides as well? One sec. This is my computer protesting that it's middle of the night for both of us. So come on, wake up. All right, what if I do it manually? Okay, manually doesn't work either. Interesting. Next. I hope I don't have to do that for every single slide. Oh, prime the pump. Okay, so now it works. So this is what nature gave us. The tree of life. This is the doodle from one of the Darwin's letters. Very, very famous. This is what we've got. What people like me are trying to do is this. We acknowledge that there's biology, there's certain molecules that make life. What we want to do is we want to go all the way back to the origins, not literally, we don't actually make protocells. We want to kind of look at life on the very molecular level and say, what happens if we try to branch out? Some of you are probably more on this side. Oops, my pointer's kind of glitchy. Wake up, it's morning. Okay, most of you are probably here. You want to make something that is lifelike, but does not come from the same chemical or conceptual lineage. I will not be defining life in this talk. I know a lot of people are, during this talk, probably someone at some point will ask me, how do you know you've done it? What is life? No, we're not going to have those. We can have those conversations over beer, but it's too early in the morning to have those conversations right now. What I will be talking about is a very specific iteration of life like chemistry. And so what I do is I'm here trying to make lifelike systems that resemble natural life, come from the same or similar building blocks, but are not exactly living cells, are not the living cells that came from the same lineage that every single one of your cells came from. Because when you think about it, all life on Earth shares the same chemistry. And if you have any training in chemistry, you probably recognize that life is incredibly boring from the chemical point of view. I mean, there's only five types of nucleobases. There's only really 22 proteinogenic amino acids. That's not much. When you think about this whole diversity of molecules that is available and the diversity of form and function, possible form and function, that you could imagine a life to realize, the biological life really is pretty narrow. There's a lot of roads not taken, never born biopolymers, however you want to call it, things that life could have done but never bothered to do. And this is what we're doing. This is kind of where we are. We're trying to make lifelike cells. So for one, we can explore what we could do, what's possible to do, but also so we can understand biological life better. So then we can modify it, we can study it better, and then we can start really calling ourselves bioengineers. Because what I'm doing often is lumped with bioengineers. A lot of the kind of communities that we hang out with are the bioengineering communities. And most of the people that do call themselves bioengineers are really cheating, because you cannot be an engineer if you don't understand every single component of the system you're working with. And biologists don't. Even the simplest minimal living cell, the Craig Venter's JCVI mycoplasma-based synthetic cell, there is, I think the current going number is 40 something in low 40s of essential genes of unknown function. So genes that we know are necessary for life, but we have absolutely no idea what they do. We have the full sequence of that gene, but we don't know what those genes do. So how can you call yourself an engineer if you're working with a chassis that you don't really know what it does? It's kind of like trying to fix an airplane by randomly screwing and unscrewing random things and hoping one of those things will help, because you don't really have the whole blueprint. That's what biology is right now. We don't have a blueprint of a cell. So what we're trying to do in my field is we're recognizing that, okay, we don't have a blueprint of a cell, A living cell is a black box. So instead of trying to poke in the dark, let's make our own cell. Let's make it from scratch. Let's make it from completely controllable components, so we actually know what we're doing. So there's kind of the two ways you can approach this problem. One is here, purify your molecules, put them together, and so you know every single component, but it's an incredibly simple, limited system. The other approach is the biology. So you have this incredibly complex living cell, but you really don't know where every molecule goes. You don't know what every gene does. You don't know how all the pathways are arranged. So what we're doing, what my field is doing, is we're kind of trying to find a happy middle. We're building those synthetic minimal cells. They're not really synthetic and they're not that minimal and they're barely cells, but we're calling them synthetic cells because they're cell-like objects that are made from controllable molecules. So we know exactly what we put in there, and because of that, we can control them. I want to make it very clear that I don't think the synthetic cells are yet living, and I'll show you some data in a few minutes that shows you kind of where the field is right now, how far we managed to go. This is not yet live. I'm kind of cheating my way into this conference too, because we haven't made life yet, but we make lifelike objects that hopefully one day everyone will agree are living. But in order to make that synthetic cell, you can kind of imagine taking those two different approaches. One is you go bottom up. So this is the bottom, purified molecules, and then you go up, which means you're building up complexity. Or you can go the so-called top down, where you take this very complex system that's undeniably alive, but we don't understand it. And we try to strip it to its elemental parts until it becomes clear, simple, and understandable. These are kind of two competing approaches in our field right now, and makes me very happy that recently there's been this so-called middle out approach emerging, which I strongly believe in, which is let's not think about this top-down versus bottom-up nonsense. Let's just focus on making the cell using whatever parts, whatever functions, whatever techniques we can. And that's why it's called kind of a middle out, because it joins this very simple chemistry, molecular biology of putting molecules together into a lifelike system, but also draws from the knowledge and methods of molecular biology. And to me, at least, I think that's the pathway to making an actual living cell is let's just not bother with kind of being purists of techniques and so on. Let's just make something that looks like a cell and let's be happy with it. Let's study it. Let's call it a cell. So what is a synthetic cell? I'm not going to give you one good definition because there isn't one. But to me, a synthetic cell is something that has not been alive before I start experimenting on it. So when you think about it, all life on Earth, yourself, the bacteria in your gut, your dog, everything we see alive around us right now came from an unbroken line of life that's been around for about 3.8 billion years on this planet. There has never been, as far as we know, another origins event that would make it till today. So everything today is a descendant of another life. There's no life just sprouting out of nowhere. And so what we want is we want to break that cycle. We are using biological molecules. So in a way, we're not completely divorced from biology. But the biological molecules that we're using are stone dead when we get our hands on them, and we put them together into this lifelike system. So basically building something that resembles a living cell, but is not coming from this unbroken lineage of life makes more life that makes more life. That's one reason why most people in our community don't think that the JCVI cell is a true synthetic cell, because they did not start with a dead cell. The genome transplantation, the way they make that synthetic mycoplasma cells, they did a completely synthetic genome, but it's been transplanted into a living chassis. And so that makes it not a spontaneous generation, not spontaneous, I don't know, grad student intervention doesn't count as spontaneous, but not a man-made directed generation of a living cell. So what we want to do is we want to actually make a cell from scratch, from non-living components. And this is as close as I can get to a definition. Something that has emergent properties. So it's more than just a sum of molecules. So it's not just a bag of molecules where every molecule does what it's assigned to do and there's no emergent properties. We want emergent properties because life is an emergent property of chemistry, but we want to be able to control it. And that controllability is really what drives the field. We want to control everything. And really, we want to control the things that you cannot control in a normal living cell. So things like, for example, size of a cell. You cannot take an E. coli, blow it up by order of magnitude and expect it to still live. You cannot take any living cell and completely rewrite its genetic code and expect it to still live. You cannot expand the chemical diversity of life. You're stuck to the amino acids that you're able to translate in natural cells. What we want to do is we want to eventually move beyond that. We want to have this lifelike system, but we want to use all kinds of chemistries that don't necessarily resemble what nature uses today. And so that's the rationale for our field. That's why we're really excited about it, is because we want to be able to control all those chemical properties and hope that this translates into just being able to control properties, what kind of enzymes we make, what kind of molecules we make. And this is what, when I say synthetic cell in my lab, this is what we mean. It's a liposome that right now follows a central dogma. So on the previous slide, I said we want to control everything. We're not that good yet. We are still using mostly canonical molecules. So there's DNA genome, there is protein expression done by the ribosomes. Right now, they're mostly just vanilla bacteria ribosomes. We are doubling into modifying ribosomes. So right now, some of the ribosomes we're having are not the natural ones, but mostly they do look like ribosomes. They still have the peptidotransferase center made of RNA. We're using RNA molecules for all the things that RNA is supposed to be used for. So we have tRNA, we have mRNA, we have some regulatory elements, small molecules. We have amino acids. This is where the field made the most progress so far in divorcing ourselves from natural biology, which is we are able to use a whole variety of unnatural amino acids. And then we have a compartment. We're building cells, and a cell is supposed to be that little cell that monks live in. So it's supposed to have a compartment. So, and that's what we want. We want a compartment. That's not the only way. There are some people that make biological cell-like structures that don't really have a membrane, but in most of my work, we do have a membrane for practical purposes mostly. And so that membrane is mostly made of lipids, just like your own cells. Your own cells are made of phospholipids, a lot of proteins and cholesterol. That's what our natural cells are made of. In synthetic cells, we're using much simpler membranes, but they're still mostly phospholipid bilayer membranes with some cholesterol in it. For some more kind of a prebiotic-like experiment, we're using free fatty acids as our membrane building components. But generally, it's just the well-behaved unilamellar bilayer membrane. So it's basically two kind of leaflets of lipids that assemble into a vesicle. It's essentially a soap bubble. When you're blowing soap bubbles, that's what you're making. You're making that kind of a layer that supports itself on hydrophilic interactions, and that's what my vesicles are, tiny soap bubbles. That's not the only way. We also use droplet microfluidics. I don't like droplet microfluidics, one, because I don't know how to do it myself. I have to rely on other people to do it, but also it doesn't have a membrane. What kind of cell doesn't have a membrane? But for some experiments, especially if you want high throughput, high kind of a resolution, very uniform compartments, we can do droplet microfluidic experiments. The really interesting stuff is what happens inside. So the metabolism of the synthetic cells, this is the chemistry playground. This is what we put inside it. So we call it cell-free protein expression system. So again, we're not completely free of biology yet. We grow bugs, we kill them, grind them up, extract the ribosomes, all the other components, and put them back together into a reconstituted transcription translation system. So this is what biology does, but we do it with purified, well-controlled molecules. So we actually have a control of every single chemical that goes into this system. And so why are we doing it? What is this good for? So this is going to be kind of a preview slide. I'm not going to show you data for any of those cool applications, but this is why people are excited about this field. One is we're making synthetic cells so we can better understand natural biology. And there is this awfully overused quote from Richard Feynman, which I actually love because it's very accurate. What I cannot build, I cannot understand. And that's true. We cannot think we're going to understand biological systems completely unless we actually can build them from scratch. So building synthetic cells helps us understand natural biology. It also helps us build tools for studying biology. If any of you ever had done any time in molecular biology lab, you probably used a technique called Gibson ligation. This is like the most common cloning technique right these days. And Gibson ligation was invented by Dan Gibson when he was working at JCVI on making the first mycoplasma-based synthetic cell. That shows you this feedback loop. We need tools to build life from scratch. We need advanced molecular biology tools. But once we make those tools, then they spread through the community. The other people start using them. They just become general use synthetic biology tools. And so this tool development is kind of the other thing we really like. Metabolic engineering that kind of falls into tool development. If you have your metabolism built from scratch, you're able to modify it. And we really need to start modifying living cell metabolisms if we want to start making compounds that nature doesn't usually make. These things that, you've probably all heard about how bad oil is for us and how we have to switch our economies away from oil. Those chemicals are too toxic to make with natural cells. But if we engineer a metabolism, we'll be able to make petrochemicals, other chemicals that our economy needs with biological or biology-like compounds. So this is this kind of a metabolic engineering angle to this. Other examples of tool development, things like microscopy, for example, to image those kind of a little cells, we need better ways of super resolution microscopy, better ways of immobilizing liposomes, which also translates to better ways of immobilizing cells. Another kind of a technology that came out of our field, there are two that came out of our field that are used widely right now. One is nanopore sequencing. If you heard of the DNA sequencing machine that's up on the ISS, that's a nanopore-based DNA sequencing machine, the mini-ion. That came out of the origin/synthetic cell research. That pore protein that was first used to build the nanopore sequencing came out of Dave Deemer's work on origins and building synthetic cells. So there is this kind of a feedback. Another one is, I hope all of you got COVID vaccine, and most of you probably got the liposomal vaccine. The liposome technologies that were used to make that vaccine a lot of them came out of this liposome research field that also feeds making liposomes for synthetic cells. So what we do is not just kind of a cool scientific questions, but it also does provide tools that have actual useful applications. Diagnostics and therapy, synthetic cells have been demonstrated to do things like shrink tumors in living mice that work out of Avi Schroeder's lab at Technion. They can promote vasculature growing organelles. They can make vaccines. They can make pharmaceuticals under very resource-limited conditions, which I'll get back to in a second. But there are actual biomedical applications. This is really that tiny little corner here is most of what's funding mine and a lot of labs in our field right now, because there's money in biotech, there's money in drug making, and we can take a little bit of that and use it for the basic research that's actually fun, but this is the useful stuff. Another thing is biocomputing. So all of you probably heard of this big promise of making biological computers that never happened because cells have a mind of their own and they do first the things they need to do to survive. Well, synthetic cells are stone dead. They don't have a mind of their own because they don't have this evolutionary baggage. We can actually make biocomputing circuits with synthetic cells without all that noise. Space exploration, long-term human space exploration requires low-cost, on-demand biomanufacturing techniques under very low-resource environments. This is perfect for making biomanufacturing solutions with things that can be lyophilized, stored at room temperature, which is synthetic cells, and I'll get back to that topic in a second. Origins of life, obviously, we're making very simple lifelike objects from scratch. That helps us understand the origin of life. And then something that's really important, and I don't know if I'll have time to really talk about it, but what we're doing is the ultimate gain of function event, and we should not be shy to say that. And so we really have to consider safety security considerations when we're doing that work. Okay, so that all sounds really cool. I hope that all sounds really cool to you. It sounds like, we're going to change the world. This is super cool. Why have I never heard about it? This is crazy. This is never going to happen, right? Well, I want to show you a quote from an article published in New York Times a little over 100 years ago. This article, I swear that becomes relevant in a sec. This article was about the feasibility of building an airplane. So building a flight heavier than air. So beginning of the 20th century, we have dirigibles, we have balloons, humans can fly, but every machine that we built up to that point is lighter than air. And so the common engineering wisdom was that in order to build a flying machine, it has to be lighter than air.
And some crazy people were saying it's actually possible to build a flying machine heavier than air. Well, that's ridiculous, right? The very respectable journal, New York Times, published a very long, very well-argumented article that says this is absolutely nuts, you should never be trying. And then 2 months later, the Wright brothers built the first Wright Flyer. The reason I love this kind of a comparison is I'm telling you we can build life from scratch. There's this whole field of people that are working on building living organisms from scratch from well-known molecules. And if you think this is nuts, you're not alone. A lot of people think that, and they're not shy to tell me that. But I will tell you, if you tell scientific and sciences and anyone in the fields of science, engineering, that something is impossible, we will just tell you, okay, wait a second, we're about to do it. And so that's what I want to show you in the next few minutes, is I want to show you where we are with this technology. So how far we actually came in putting together lifelike systems from scratch. So what's the hallmark of life? Like when you think about life, what comes to mind? I'm not going to actually drag the audience to answer that question. It's too early. But when most biologists think about life, what they think about is, well, those black zoomy things are really not helping anyway. Trust me that what it says is a complete cell cycle of an evolving synthetic cell. That's the slide title. And that's what we did. We made a synthetic cell, so a completely controllable chemical system that can undergo a complete cell cycle and evolution. And so when you think about, if you're a biologist, when you think about hallmarks of life, cell cycle is one of them. In order to call some chemical system alive, it has to undergo a cell cycle. And so that's what we did, is we built a synthetic cell that's capable of undergoing a complete cell cycle. And we demonstrated that it's possible to have natural selection in those systems. So what we have is a compartment with liposomes. Again, I'm a lipid person, so these are liposomes. And then inside that compartment, there is a genome. That genome is on few plasmids, and I want to point out the size of that genome. So SYN 3.0 is that most minimal mycoplasma-based synthetic cell. This is the simplest independently living organism right now. And it has a genome of the size of about 500 kilobase pairs. Our genome is 90 kilobase pairs, 90. There's been theoretical work that predicted that the absolute simplest independently living cell should have a genome on the size of about 120 kilobase pairs. So we're getting there. Our genomes are already approaching the size that is compatible with independent life. So just to give you kind of a scale, this is not a toy model system with a couple dozen genes anymore. We actually have genomes that are pretty respectable in size. Then we have replication of that genome. This is what you need in order to maintain cell cycle, in order to evolve. So we're replicating our genome. Then we're expressing our genome. We have protein expression system. This is based on central dogma. So this is what cells do naturally. We have ribosomes that express proteins out of that genome. Then those proteins, some of them go into the membrane, and they go into the membrane for a few reasons. One is because we want to have waste removal and nutrient uptake, communication with the environment. So basically, a lifelike system cannot be closed from the environment. It has to be able to exchange matter and energy with the environment. And so in our case, those membrane channels allow us to do that exchange. Now, you have to be able to grow if you want to think about lifelike properties. And so this is the first example of a system where growth of synthetic cells is genetically encoded. So our cells grow as a result of protein expression inside the synthetic cell. So that couples the genotype, so the gene that encodes that protein responsible for growth, with the growth itself. It sounds trivial because all life does it, but that has never been done in an actual fully controlled chemical system like ours. So we can express a gene that induces growth. Now, what happens when you grow? You can grow a little bit or you can grow more. And that we can control, because in our system, and I'll get into details of that in a second, but this those kind of two different cells, one, we call them T7, I'll explain in a moment what it means, but that cell grows less T7 max cells grow faster. So basically think of it as two different populations. One of them grows slower, one of them grows faster. Here's your beginning of a natural selection. One population grows slower, the other population grows faster. The population that grows faster grows bigger. And in most of the animal, plant, bacterial world, if you grow bigger, you can produce more offspring. That's sadly not true with humans, but it's true with most bacteria. If you're really big, if you grow fast, you can produce more offspring. And that's what happens. This is the reason why I don't think this is really living system yet. We don't have spontaneous division. And I'll talk about that in a sec. We divide manually. So to divide our cells, we have to literally push them through a little filter. We call it extrusion. We push them through a filter with little pores, and that creates daughter cells. For some reason, it's always daughter cells. That's what everyone in the field says. There's no son cells. When you divide liposomes, when you divide cells, they're always daughters. So we make a lot of daughter cells, and the ladies that come from the really big mother cell, there's more of them because they all get divided to the same size. So this is your natural selection. The guys that were small to begin with, the ladies that were small to begin with end up with less offspring. And the really big fat ladies end up with a lot of offspring daughter cells. And this is how this genotype gets coupled to the amount of offspring. If you grow a lot, you produce a lot of offspring cells. And I'll show you data for that in a sec. And then last but not least, we have what we call a generation counter. I'm not going to go into details of that. It's basically a way to prove that the lineage remains consistent. So one and the same synthetic cell went through cycles and cycles of growth and replication. Okay, so I want to show you now some data that I'm not just making all that up. I'll show you an actual data to prove that this is happening. So this is what we did. We did generations of experiments. So you have your starting population of synthetic cells, you feed it, it grows, you divide it, feed, grow, divide, so on and so on and so on over days and days and days. These are kind of annoying experiments because there's no stopping point. You cannot freeze it, you cannot put it on a shelf, you actually have to be there every, so that the full cycle is 12 hours. So you really have to be there every 10 hours to start defeating an extrusion. You can imagine favorite projects for my grad students. I've done some of those experiments too. I took some middle of the night data points, so I'm not embarrassed to say that. But yeah, this is kind of annoying that they're very princessy, like you have to feed them, you have to divide them at a certain time. That's kind of why I would love spontaneous division, so I can set it up and forget. We don't have that yet, but we divide them by hand. Okay, so now I'll show you some data at generation 5. Why generation 5? We did do more generations, but the kind of a quality of our analysis, data analysis goes down after more generations for many reasons that I'm happy to talk about later. But basically what I'll show you now is data for after 5 generations. So we did this 5 generations of growth and division. Now, data warning, the next few slides will be kind of a hardcore molecular biology data. If you don't like that, go get coffee. Okay, so what we do is we have our genome. So the first thing we want to see, so what you see here is qPCR data that shows you that we have new DNA. So remember, there was genome replication as one of the steps of our cell cycle. So we start with a particular genome, and then we have a way to get rid of that original genome and only quantify the amount of DNA from the new genome. So the genome that's been synthesized in the reaction during that cell cycle reaction. And that's what we quantify here. So we have, yeah, this is really annoying that those squares. What I want to show you is these are experimental conditions. And so what you really should focus on is this guy. This is the complete cell cycle. So this is DNA replication and feeding. And this is our negative control. So this is no DNA replication and no feeding. And these are the 2 conditions in between where we either replicate DNA but don't feed them, or don't replicate DNA, feed them. But really the biggest comparison is between this first one and this last one. So first conditions, this is the full DNA replication and feeding. We have good amount of new DNA for both plasmids. We actually did those experiments for every single plasmid in our genome. I'm showing 2 representative examples. So every single gene on our plasmids does get replicated, produces new DNA. Now, is there mRNA? This was the first surprising example. So again, what you see here is quantification of mRNA from the genome. So we have those genes, they get transcribed into mRNA, we can quantify that mRNA. What I would expect is if I have those cells that grow and get fed, there will be significantly more mRNA than in the cells that don't grow and feed. So if I compare this guy with that guy, there will be significantly more mRNA here than here, and there isn't. And I was driving me nuts for a few months when we first started doing those experiments because I thought something's wrong. Nothing's wrong. The system already has emergent properties, which is fantastic. What happens is there is a competition between the enzyme that tries to replicate the DNA. So that's the 529 rolling circle polymerase that's replicating our plasmids, and the RNA polymerase, the T7 RNA polymerase that makes the mRNA. And that competition results in this kind of equilibrium that we're reaching, where if we have a lot of genome replication, then we have less mRNA because the occupancy of the T7 RNA polymerase decreases on that template. And this is a very simple system that the dynamic already emerges if you have just a few plasmids. It doesn't have to be this whole 90 kilobase per genome. And so this is really cool because this is the first example where what we're building is not just a simple chemical system that behaves like the molecules separately, but the molecules start interacting with each other, having those kind of a cool emergent lifelike properties. So next thing is we look at protein expression, and this looks a little more like you would expect. The proteins accumulate. This is because proteins accumulate while mRNA gets degraded. Proteins in our system do accumulate over generations. We don't have any protein degradation systems. So here's an example of a green fluorescent protein that we make in the system, and it keeps accumulating over generations. So the ones that do get fed and divide do get more protein over time. Last but not least, this is a technical, but I'm obligated to show it. Generation counter demonstrates, and I'm happy to talk about the mechanism of that during the break, but that generation counter basically tells you that every cell in that system does go through growth and replication, in this case 5 times. The fact that we have any detectable signal in those, so here, that means the generation counter shows that we did go through 5 cycles of growth and replication. Okay, so what we have here is genome that gets replicated, proteins that get expressed, cells that grow, and then go through generations after generation after generation. And I promised that I'll talk about some sort of an evolution natural selection competition. So this is actually my favorite part of this project is we weren't expecting to see that at the beginning when we first started that, but we do see kind of an emergent dynamics in those systems already that have kind of a lifelike, I don't want to make too many claims, but properties that resemble natural origin of kind of a very beginning of natural selection. So what's this system based on is, remember I said T7, T7 max. These are the names that these are kind of random names we assigned to 2 different, again, those black thingies are blocking what I really want to show, but the way the fusion system works is we have those membrane proteins that induce growth, and those membrane proteins can be under a weak promoter, which means you get less protein, or a strong promoter, which means you get a lot of protein. The weak promoter is called T7, and if that sounds familiar, that's because that's the same T7 RNA polymerase that makes the mRNA vaccines. It's a great polymerase. We engineered a new promoter for that polymerase that we call T7 max that significantly increases expression from that promoter. No protein engineering here, just engineering of the sequence of the promoter. And we call that promoter T7 max. So with the same gene, the same T7 RNA polymerase, the genes under the control of the regular wild type T7 promoter express at lower levels than the ones under T7 max. And here's a flow cytometry that shows you that with the T7, so this peak is what you really should look at. If someone wants to talk experimental details, I'm always happy to, but the take home message is that this peak is smaller than that peak. And that basically indicates the yield of the fusion, that indicates the yield of the feeding. So the ones that have their feeding protein under the T7 promoter grow slower than the ones that have their feeding protein under this stronger promoter T7 max. Where am I going with this? Well, this is our fusion experiment. So this is after 5 generations, 2 different conditions. DNA replication, feeding, and growth. So this is the complete cell cycle, or nothing, no DNA replication, no feeding, no growth. We look at how many cells we have if we let 2 populations compete. So we're starting with equal amount of the wimpy T7 ones and the strong T7 max growers. We're starting with equal amounts at the beginning. I make them from scratch so I can decide the original ratios. So we mix them 1-to-1. This is what those blobs here represent. The light gray ones are the weak T7. The kind of the darker ones are the T7 max. And if we put our GFP reporter, so the GFP is a fluorescent protein that lets us count those cells. If we put our GFP reporter in the cells that grow faster, we can see that there is more of the ones with GFP than the ones without at the end of the experiment. That's as expected. The stronger ones grow faster. Now, the opposite experiment, we just did it as a control. This is why you should never do controls if your experiment works, because that just kind of creates a rabbit hole. We did a reverse experiment. So in the first experiment that I'm showing, we put our green fluorescent protein in the cells that grow fast, and we expect that after multiple generations, we're going to have more green cells because they grow faster. So there's bigger percentage of population. And that worked exactly as expected. Now, we put GFP in the ones that grow much faster, and we see more of them. So after growth and division, we see more GFP. Now, this is the difference. So what you see here, the first 2 bars, there's about 15% less cells that grow slower when the GFP is in the cells that grow slower. And now here, there is about 9% more of the cells that grow faster if the reporter is in the cells that grow faster. So basically 2 different experiments. In one of them, the reporter protein is in the fast-growing ones. In the other experiment, the reporter protein is in the slow-growing ones. The slow-growing ones are losing, because we're just looking at the kind of a relative ratios, how many of the slow versus fast growers we have in our tube. So those 2 numbers should be the same, because one of those numbers shows you how much the slower growers are losing. The other one shows you how much the faster growers are gaining. Those numbers should be the same. They should be identical. We've done this experiment enough times that I'm really confident that the error bars are much slower than our actual result. So this is the same data just plotted in a different way. There is about 15% more of the fast growers after 5 generations if the reporter is in the slow growers, and there's about 9% more of the fast growers after 5 generations if the reporter is in the fast growers. Those numbers should be the same. That was driving me nuts for a really long time. Why are those numbers not the same? The slow growers are losing their relative kind of occupancy in the population at the same rate as the fast growers are gaining. There's just no physical way for any third population of cells to arise in there. So what's going on in there? So what we did is, so in those experiments, we only were tracing one population at a time because we had one fluorescent reporter protein. We repeated those experiments, but this time we're tracing both populations. So both of those populations now have a reporter protein. One of them has a green reporter protein. The other one has a red reporter protein. And suddenly those numbers started matching. there's about 10-ish% difference between the slow growers and fast growers. And first that made me super confused, and I was thinking about it for quite a while, but then it made me extremely happy, because what we see here is an emergence of a very simple regulatory mechanism that's related to the resource availability, a chemical resource availability in that system. What happened on this side in the first set of experiments is that if we put that additional reporter protein, the ones that grow faster are now a little bit distracted, because in addition to growing, they also have to spend resources on making that reporter protein. If we shoot both of them in the knees, so both populations have to now make a reporter protein, suddenly the slow growers lose as much as the fast growers are gaining. And so this is a simple system. This is only 90 kilobase per genome. They don't make thousands of proteins. They only make about 120 different types of proteins. But already forcing them to make this one type of a reporter protein creates differences and kind of induces this regulatory mechanism. I'm hesitant to call it homeostatic mechanism, but it is kind of a very simple way of regulating your metabolic rate that's related to the burden, the metabolic burden in your population. So this is dead as a doornail. This is not a living system. This is just chemical bioreactors pretending to be cells. But we already see those emergent properties that allow us to say there's something going on that's more than a sum of its parts. So we see emergent properties in this very simple cell cycle, this very chemical system that undergoes a cell cycle. So again, what we have is we can do 5 cycles of growth and division. We can do more, but I'm showing data for 5. We have emergent properties. We can undergo this kind of a cycle of growth division. So I promised I'm not going to discuss definitions of life, but I'm obligated to put that on the slide. NASA definition of life is self-sustaining chemical system that's capable of Darwinian evolution. That's kind of it's a crappy definition, because according to that definition, neither of you is alive, but it's one of the few definitions that we're kind of having practical use. So this, according to this definition, those synthetic cells that undergo a cell cycle are not yet alive because they're not self-sustaining. And that's why I don't think they're alive. But that's why you also can have a discussion, because this is a really crappy definition of life, because no life is truly self-sustaining. No life is truly self-replicating either. I cannot replicate without my husband, so I'm not alive if self-replication is requirement for life. So that kind of shows you, one, why there are no good definitions of life, but also why some people might think that if you have this complete cell cycle that enables competition, it could already start crossing into life. So why I don't think it's alive? Because this is not a true Darwinian evolution yet. Those guys that compete, this really cool emergence of homeostasis mechanisms, that was inserted there artificially, because we put in there those fast growers and slow growers. Those mutations do not arise spontaneously. So for me, for this to really try to become a living system, the mutations have to be arising spontaneously. And that's my personal definition. You can crucify me for that. That's kind of what I believe, is that you need to be able to evolve on your own in order to be called living. Okay, so this is the cool part. Now I want to switch gears for the last few minutes just to show you a little bit of useful but also cool stuff. So one is you have this cell-like system. You can completely describe that system with a list of molecules. So what happens when you can completely describe a composition of a biological system? Well, you can send information about that biological system to a remote location, and they can recreate your experiment without the need to physically send samples. Well, if I'm working with a collaborator in Europe, then I can just send you a FedEx sample. It's obviously faster if I send you an email, but it's possible for me to send you a physical sample. But if my collaborator is somewhere farther away where FedEx doesn't deliver, then I cannot send you the sample anymore. And this is the real power of this technology. We can send information about making biological systems remotely, as long as we've previously agreed on a set of or set of building blocks. We all have those building blocks. You should be able to recreate my experiment from the recipe I give you. This is not possible with biology. If I make a new strain of E. coli, I have to send you that strain, unless you happen to have a parent strain and you can reproduce that whole evolution experiment. So here we can have building blocks, chemical building blocks, and we can recreate each other's experiments. What is it good for? Well, this is obviously super cool, but it has practical applications. And one of those practical applications is making drugs on demand in low resource remote locations. So when we send Elon Musk to Mars, hopefully he'll stay there, but he will probably get sick at some point. So we want to make medicine for him. So how do we do that? We cannot plan in advance. How do you make medicine? What medicines are you going to need? But you can build a biomanufacturing platform that will allow you to make compounds on demand. And so this was the premise of this project that we call astropharmacy, where we use synthetic cell chassis there's this idea that yes, we can use this to make medicine on demand, we can make small molecules on demand, but there was a question, does it actually work under spaceflight conditions? So I was lucky to be a part of this amazing group of people that set out to test that. So these are people that are mostly interested in making drugs. I like drugs like everybody else, but I'm more of a kind of on the technology development side, on the development of the actual platforms to make them. So what we did is we asked a question, is it possible to do two reactions under spaceflight conditions? One is a transcription of a functional RNA. I really don't have time to kind of go into details of that, but this is that one of those squiggles is a functional fluorescent RNA aptamer. The other one is a short peptide. So can we do transcription and translation from a lyophilized protein expression system? So that's the cytoplasm of a synthetic cell that we can lyophilize, store at room temperature. Can we do that under spaceflight conditions? Honestly, the main reason I'm showing it is to show you this video, because that was the first time my own sample went into space. So I was obviously super excited. I have to be up at 4 a.m. to watch that launch. So now I'm torturing everyone with that video, but it did work. The samples had lyophilized material. It was sitting, the launch was a little bit delayed. So it was sitting on a tarmac in Virginia for 2.5 days in the middle of the summer. So temperatures were above 100, that's above 30 in the normal degrees. And so it was sitting there, it was hot, it was then shaken up on the way up, and it works. We can make medicine under spaceflight conditions. So we can, we haven't actually shown we can make medicine, but we can make proteins. So making small molecules should be next step. So what we're doing now is we're sending some of those experiments up to the ISS to actually have people doing the pipetting. That was an automated experiment. Now we want to do the pipetting to actually show production of 3 different small molecules that actually have some biological properties. But the take-home message is that those systems are not as wimpy as they look. You can freeze dry them, you can shake them, you can send them up to space. They will make proteins, they will make functional RNA after that long time. Okay, so a lot of cool applications that I told you about. I want to spend the last remaining couple minutes to talk about something that is really important, but often not discussed in our field. What we're doing needs to be done in a safe and secure way. We're building technologies, we're building capabilities that, one, have never been available before to life in general, and also provide ways to kind of circumvent a lot of safety and security safeguards that are in place for biological research right now. So what we're doing is we're not just kind of running with it enthusiastically, but we're also asking what can go wrong. And so we're doing this so-called horizon scanning for technologies in our field specifically and in engineering synthetic life in general, which is what can go wrong? If you do a mouse analysis of particular synthetic cell experiment, what can accidentally go wrong and what a possible bad actor can do to make it go wrong. And this hopefully will help us build safeguards to do those experiments in a safe, contained way. And some of the things that we're doing is we're working with the regulatory agencies, we're working with all the possible stakeholders, we're working with the scientific community, but we're also working with the public. There is a great citizen science movement. People are literally they call it biohacking, they're doing biological experiments in their garages. And this is a great platform to use for that because it's relatively safe. But when you're doing it, when you're spreading it out into the wild right now, we have to think about safeguards, what can go wrong, how can we do that in kind of a responsible way. And then obviously the taxpayers that pay for our research, we know what's the scientific literacy in our society, and that's sadly lower than we would like. So we're using those kind of the platforms to demonstrate the benefits of it, but also to talk about why we're doing it, why is it good, and why is it safe. And last but not least, there's literally dozens of us. That's, I want to plug the Build-A-Cell community here. A lot of you probably think you don't belong in the Build-A-Cell community because we're the biologists, chemists, and you're doing something that's not biological or chemical, but that's not true. We all want to make living organisms from scratch, but in order to do that, we need expertise of people from all different areas. So what we want to make here is, just to be very clear, we're making biological lifelike systems, but we're making those systems for not just for basic research, but also for practical applications for biomedicine, for metabolic engineering. And we're doing it in a way that kind of needs this whole international scientific community. And Build-A-Cell does not define life. This is one of the things that I feel really strongly about, is that we should not limit ourselves to any one particular design. The main thing we do is we bring people together, we drink coffee, eat cookies, and work. We have workshops in person. That's a really big part of our community, is we have workshops twice a year in person. We actually all come together, meet in person, sit down. There's no talks at those workshops. We actually literally sit on our butts and work all day. And we do that twice a year. Working groups come out with white papers, publications, policy, things that can be used to produce new regulatory recommendations, but also funding recommendations. We're working with funding agencies, but we're also working with citizen science communities. We're working with the general public, and shout out to the National Science Foundation for funding us. And I want to say thank you very much to all of you for having me here. I want to thank everyone in my